Good morning again. I'm Mark Roithmere. I'm CEO of the Alzheimer's Drug Discovery Foundation. I'm delighted to be with you today at my first Fall Luncheon and Symposium in this role. Today's symposium perfectly exemplifies the impact of ADDF and our investments. It features 3 brilliant researchers who have developed novel drugs and reached clinical trials. Very exciting. As you will learn, that represents a lot of hard work, and it couldn't have happened without the vision and leadership of our founders, Leonard and Ronald Lauder. And it is now my honor to introduce our co-chairman, our co-founder, Mr. Ronald Lauder.
Good morning, and thank you, Mark, for that kind introduction. It's my pleasure to welcome you to the Fall Symposium. Today's panel is led by ADDF's Executive Director and Chief Science Officer, Dr. Howard Fillit, and it showcases just how far we have come in our mission to conquer Alzheimer's. My brother Leonard and I started ADDF almost 20 years ago. There was almost no hope for Alzheimer's patients. We wanted to change that. Alzheimer's patients need effective drugs to treat this disease, and people at risk for Alzheimer's need drugs to prevent it. We created this foundation to find the best ideas for new drugs and to help them advance in clinical trials. And when we hired Howard Fillit, we were thrilled that he was able to join us and lead these efforts. The 3 panelists today each have a great idea. ADDF gave them the financial support to develop these ideas. They developed these ideas into drugs to treat Alzheimer's, and they did develop them. All 3 of the scientists here today have drugs that have reached clinical stages. Getting a new drug to clinical trial takes a decade or more of tremendous effort, and they did it. Most people don't make it, and we congratulate each of these people for getting this far, and we encourage them to keep forging ahead. There today are only about 130 treatments for Alzheimer's in clinical trials. That's not a lot when you compare it to other diseases. For example, listen to these numbers. There are 1,500 treatments for breast cancer, 750 for heart disease, 600 for diabetes, and only 130 for Alzheimer's. Alzheimer's is the only disease of the top 10 without one proven treatment. And unlike breast cancer, heart disease, and diabetes, there are no survivors. But the number of drugs for Alzheimer's is growing, and that is due to the hard work of Howard Fillit and everyone at ADDF, and also donors like you. In fact, 20% of all the treatment in clinical trials for Alzheimer's have received ADDF support, and more on the way. After the symposium, we invite all of you to join us for lunch. This year, we are honoring the remarkable Sharon Sager for her support of Alzheimer's research. Now, it's my great pleasure to introduce Howard Fillit and our panelists. Howard?
It's a great pleasure to be here today, and I want to thank everyone for joining us today. It's a wonderful thing that you have. We have a great group of researchers with us today on the panel. As Ronald mentioned, each one of them have taken a drug from preclinical chemistry and brought it into human clinical trials for the first time. Each one is targeting some aspect of what is called synaptic function. And because this is really the focus of what they're doing, and I want to spend a couple minutes telling you why synapses are important and what they are. And we thought maybe we'd start with a little video to show you a little bit about how the brain works, which is obviously quite complicated. So this is your brain, and as we drill down, the neurons obviously are the brain cells that create enable us to think. And in the brain, these neurons communicate with each other through synaptic tasks. And there are literally trillions of synapses in the human brain. And we know that Alzheimer's disease probably starts at the synapse. Some of the plaques may even start at the synapse. So dysfunction of the synapse is critically important. And it was shown many years ago that there's a direct correlation between synaptic loss in Alzheimer's disease and cognitive impairment. And so it's always been thought to be one of the most fundamental problems in the disease. If we could, I just want to say I'm using this teleprompter for the first time. It is a little confusing. Can we move forward? Okay. Yeah, let's keep going. Okay, so as we learn, what happens is that the synapses actually grow stronger and they reinforce themselves. So actually the basis of learning is actually at the synapse. As we learn something, we form more synapses, and the synapses that we have become stronger in many different ways. Okay, let's keep going on that. I'm sorry, I haven't done this before. Keep going. Where's my friend up there? Okay, I've already said that. I guess I memorized this thing, and okay. Yeah, I don't need a teleprompter. I know the story. Okay. Yeah, keep going, sir. I forgot your. Okay, keep going. Sorry about this. Okay. Yeah, let's keep going. So, okay, let's start here. So, these are our wonderful panelists. So our symposium starts today. I'll introduce Dr. Leen Kawas. Leen is the founder, president, and CEO of a Seattle-based company called M3 Biotechnology. She earned her doctorate in molecular pharmacology from Washington State University and a doctor of pharmacy from the University of Jordan. Dr. Kawas's drug is called NDX-1017, and it harnesses various growth factors to repair and regenerate synapses and to preserve cognitive function and slow down Alzheimer's disease. Since the inception of her company, ADDF has given Dr. Kawas several mission-related investments, and we are currently supporting the development of NDX-1017 with $1.4 million in funding to enable her drug to be tested for the first time in human beings. Indeed, Dr. Kawas launched the first phase 1 clinical trial of NDX-1017 just last week. And so it's my pleasure now to introduce Dr. Leen Kawas to tell us about the potential of her drug for Alzheimer's disease. Thank you.
A very nice introduction about the company. Before I start, actually, I just wanted to take a moment to thank the ADDF and everyone in this room, because if the ADDF didn't invest in our company and if you didn't have this mission, we wouldn't have this exciting neuroprotective and regenerative therapy in the clinic right now. So thank you, Howard. Thank you, ADDF, all the scientists that gave us a really hard time in the scientific review. You actually were very supportive and gave us really good feedback to enhance our chances for success. So let's try this. I'm going to tell you today about M3 Biotechnology. We are a Seattle-based company, and we are working on small molecule therapies with a purpose to regenerate brain cells and protect from the progressive degenerative process in the clinic. We've started the company 6 years ago, but it's based on research for over 15 years. We've hit a lot of milestones, commercialization milestones, and we are currently in our phase 1 clinical trials. We're actually in the second cohort, so we started a few weeks ago, and now it's very exciting to have the support from the ADDF for this stage. So I just wanted to show how basically the compound that we're working on works. I'm sure everyone in this room knows this. A healthy brain, when basically it retracts Alzheimer's, it goes through a progressive degenerative process. And this is a very late stage AD. You could see there's shrinkage, loss in the synapses that Howard just explains. And what we've seen from the preclinical models that we've tested is we have a preventive, neuroprotective, and potential regenerative effect of the therapy. So if we catch the disease early on, we prevent it from going to a later stage and potentially stop it when it's at a mild to moderate stage. The other exciting part for M3 in the last few years is there has been a good effort by the Allen Institute for Brain Sciences in Seattle to try and identify a new target for the brain. And this is here, they just rank ordered 30,000 genes. And one of the genes that were highlighted by their work was our target at M3. This is a brain area or region in the brain that a lot of the learning and memory happens. And this is M3's target. It's one of the highly regulated and most important targets in the brain. This is actually a study that was supported by the ADDF almost 6 years ago. And without this funding, we would have not been in the clinic today. Here, what we've tested is aged animal who show a similar memory decline like AD. Those two animals basically were trained for 4 weeks. This guy got the treatment for 4 weeks. This guy didn't get any treatment. And similar to Alzheimer's patients, there's a loss in spatial memory. After 8 days of training on this maze, they hate water, so they want to escape it. They want to try to learn anything that would basically help them to escape the water. You could see that the animal was able to escape, versus the untreated animal is still showing spatial memory decline. After that first investment from the ADDF, it was actually a long path, like Howard described. We had to understand how the drug moves in the body, how do we get rid of it. We needed to make sure that it's safe, to make enough of it to test in the clinic, and we launched our clinical trials. Along this way, we've had a lot of positive conversations with the ADDF. So it's very exciting to have an idea from the bench to an actual product that we are testing in the clinic. Today we have phase 1A and B clinical trials. We're looking to finish this next June. And this is a combination, 92 volunteers, a combination of healthy elderly and Alzheimer's patients, where we're looking at safety, but in addition, we have cognitive and biomarker endpoints. Thank you.
Thank you, Leen. We'll be asking questions and looking for your questions when all speakers have finished. Our next drug discoverer is Dr. Jerri Rook. Jerri is an assistant professor of pharmacology at Vanderbilt Center for Neuroscience Drug Development at Vanderbilt University. She received her doctorate in pharmacology, toxicology, therapeutics at the University of Kansas Medical Center. With support from the ADDF since 2011, she's been developing a drug called VU319. We began our support of the drug while it was still in animal testing. VU319 began first in human phase 1 clinical trials earlier this year. And as I said earlier, synapses are critically important for memory and cognition, and Dr. Rook's therapy is focused on a type of receptor, as I showed you those receptors in the synapse, that receives a neurotransmitter from those vesicles called acetylcholine. And her drug enhances synaptic function, making the transmission of neuronal signals more efficient and effective. And here's Dr. Rook to explain exactly how that works.
Thank you, Howard, for the introduction. I'm extremely delighted and honored to be invited to speak to you today about the development of our program and advancement of VU319 into the clinical trials. I'm excited to demonstrate our progress, and I can't emphasize strongly enough how instrumental the Alzheimer's Drug Discovery Foundation and the support Howard's given us have been in advancing our compound into clinical trials. So what is VU319? VU319 is a selective activator of an M1 acetylcholine receptor, and I'll explain to you the importance of that selectivity here in a minute. But as Howard mentioned, acetylcholine is a chemical in the brain that is essential for normal learning and memory. And this chemical is also significantly depleted in Alzheimer's patients. And so, as you can see here, in the synapse, you have a neuron or nerve cell that releases acetylcholine. It activates the following receptor, and by doing so, facilitates normal learning and memory. And this is, like I said, depleted in Alzheimer's to patients. And so, the current standard of care, Aricept, donepezil, those type of drugs, they were developed based upon this theory. And what they do is they increase the overall levels of acetylcholine. Unfortunately, this is going to be a little hard for you to see, but there are several targets for acetylcholine. They're not only located in the brain, but they're also located throughout the entire body. And so when you administer these drugs, it increases the overall levels of acetylcholine throughout the entire brain, therefore activating all of these targets and all of these receptors. These receptors are not only located in the brain, but they're located in the gut, the heart, all over the body. Overactivation then of these receptors with the treatment of Aricept or donepezil causes adverse effects, specifically in the gut, like severe GI distress. It can also cause effects in the heart as well. And it's believed that these off-target effects or effects outside of the brain are what's responsible for the lack of true efficacy or effects or beneficial effects of these drugs in the patient population, because they're not able to increase the dose high enough to have the beneficial effects in the brain while still being able to avoid those adverse effects that are seen in the patient population. And so, why are we targeting this M1 receptor? Well, the M1 receptor is highly expressed in the brain, not only just in the brain, but in regions of the brain, such as the hippocampus and the cortex, that are essential for adequate or normal learning and memory. In addition, activation of M1 doesn't have these adverse side effects that you see with overactivation of all of the receptors or the targets that acetylcholine have. And so with VU319, what we're able to do is we're able to selectively target this M1 receptor that's located in the brain. Increasing the activity of it then restores the synaptic function and then enhances learning and memory in these Alzheimer's patients where this activity is diminished. And so, as Howard mentioned, many of these programs take decades, and as an example, we began our journey almost 10 years ago, where we started screening compounds. We screened a library of over 160,000 compounds, looking for a compound that selectively activated this target, this M1 target. Following several years of development, we were able to select our lead or our clinical candidate, VU319. But now what do we do? We're here, unfortunately, what we affectionately commonly call the valley of death. At this point in time, once you select a lead compound and you're able to demonstrate that it has the beneficial effects that you desire, you then have to go through this very long regulatory process of extremely expensive safety pharmacology and toxicology studies that are required to demonstrate to the FDA that your compound is safe enough to go into clinical trials. Now, these studies are not hypothesis-driven. They're not particularly exciting studies to the scientific community, but they are essential in moving these programs forward. So unfortunately, it's extremely difficult to receive funding for these studies. Thankfully, however, the Alzheimer's Drug Discovery Foundation and Howard know how essential it is to provide funds for these studies, and they have supported then all of these safety pharmacology and toxicology studies that we needed to submit in a new investigational drug application to the FDA and fortunately, we were able to complete these studies. These are preclinical studies. We call them IND enabling because they are required to submit your investigational new drug application to the FDA. Fortunately, we were able to, with the support of the Alzheimer's Drug Discovery Foundation and a partnership that they initiated with the Harrington Discovery Institute, we were able to successfully complete these studies and receive approval from the FDA to go into human or clinical trials. And so where are we now? So now we are in phase 1 clinical trials. This is being conducted at Vanderbilt University at our Institute for Clinical and Translational Research with our collaborator, Paul Newhouse. We've just successfully completed our first cohort of dosing and have received approval from our external advisory board to go to the next step or the next dose. And so we're hopeful that by this time next year, maybe late spring, that we'll be finished with our phase 1 clinical trials. And at that point in time, what we would like to do is then advance in clinical trials into our patient population. Again, I'd like to just reiterate the importance that the ADDF and Howard and all of you have in the success of our program. I fully believe that we would not be where we are today without all of your support. Thank you.
The speaker is Mark Gurney. He founded Tetra Discovery Partners, a biotech company based in Michigan in 2011. And Dr. Gurney earned a doctorate in neuroscience from the California Institute of Technology and an MBA from the Kellogg Graduate School of Management at Northwestern. He was one of the scientists that discovered an enzyme in the brain called beta secretase, also known as BASE, which is involved in the formation of amyloid plaques. And today, BASE inhibitors are one of the leading drug candidates currently in phase 3 clinical trials for Alzheimer's disease. With ADDF support, Dr. Gurney is advancing his new drug called BPN 14770 into phase 2 clinical trials. BPN 14770 uniquely targets a system in the brain that ultimately activates genes that promote synapse formation and these neuronal connections, neuronal connections that are essential to our ability to think. And so let's welcome Dr. Gurney to explain further.
Well, thank you, Dr. Fillit. It's a pleasure to be here today. I'm really honored by the invitation to speak to you all. ADDF funding for Tetra has been critical. ADDF co-funded our second clinical trial, which was conducted in elderly subjects, and I'll show you the exciting data from that trial towards the end of my talk. And then more importantly, ADDF is willing to fund the less glamorous parts of drug discovery and development. We receive funding for critical animal toxicology studies and also manufacturing for the drug that we're now moving into human phase 2 clinical trials in Alzheimer's. So we're developing powerful drugs that will impact the brain connectome. That's the pattern of connections whereby we learn, remember, and store memories in our brain. And so three of the 4 approved drugs for treating Alzheimer's disease target an enzyme known as the cholinesterase, which breaks down acetylcholine. And you've heard from the previous speakers how acetylcholine, when it's being released at the synapse, is important for learning and memory. So our compound acts downstream of the cholinesterase inhibitors and has a complementary pattern of action. It acts directly on the synapse. It acts on the postsynaptic element of the synapse. And it affects both the earliest transient stages of memory as well as longer-term formation and stabilization of connections whereby we store long-term memory. And then also this pathway, known as the cyclic AMP PKA CREB pathway, modulates the expression of growth factors in the brain. In this case, a growth factor known as brain-derived neurotrophic factor. So BPN 14770, we just call it 770, has a complementary pattern of action to other Alzheimer's drugs and can be used as a polypharmacy. But more importantly, it has the potential to modify disease by increasing the resilience of the brain to the damage that's caused by Alzheimer's through the formation of new connections. So if we indeed can strengthen the connectome in an early Alzheimer's patient, we should delay progression. Moving earlier, and I'll show you we have memory benefits in elderly adults, we may be able to push back when the disease actually develops. So in parallel to developing our drug, we also developed what's known as a PET tracer, a tracer for imaging the target of the drug in the brain using positron emission tomography, or PET. And this very excitingly shows for the first time where our drug target is located in the brain. So this is an anesthetized monkey. Here's an MRI. This is the front part of the brain and the back part of the brain. And then this is the PET image down below, and this red speckling is where the PET tracer is binding. And what was really exciting for us when we saw these images for the first time was the realization that our enzyme target, which we thought should improve cognition, is actually expressed in the parts of the brain where we think, which are important for cognition. So prefrontal cortex and hippocampus. Hippocampus is important for the earliest stages of memory formation. Prefrontal cortex is where we store long-term memories, and then we retrieve them. And these are both the parts of the brain that are affected by Alzheimer's disease pathology. We also, using the PET tracer, can block with the drug first, and then that blocks the binding of the PET tracer subsequently. So we know our drug actually enters the brain. And then very excitingly, in our early human clinical trials in phase 1, we've now shown a cognitive benefit in healthy elderly subjects. And this is a really unique piece of data. Aricept, for example, donepezil, has benefit in mild to moderate Alzheimer's disease. It has no benefit before the acetylcholine levels in the brain have started to decline. So donepezil in mild cognitive impairment prodromal to Alzheimer's has no benefit, and donepezil in healthy elderly has no benefit. But here for the first time, we did a clinical trial where we enrolled 45 older human subjects, in this case, 60 years and older. I hate to say it, but I could have qualified for my trial. And this shows the data across the clinical trial. So here's baseline. Here's the first dose of the drug. And then the drug is dosed for 8 days, and we're doing cognition testing every other day. And this is a pool analysis. There are 20 people on active and 10 people on placebo. And we did 3 similar cognition tests in this trial. We did a whole bunch, but I'm going to talk about 3 today. And these were all sort of attention, reaction time tests involving working memory. So the first test was pretty simple. It was just you're looking at a computer screen and has a new playing card appeared, yes or no. So that's a simple reaction time task. Everyone gets that right, and there was no benefit of the drug. The second task was a little more complicated. It was either a red joker or a black joker, and you had to remember, is red the correct response or is black the correct response? And again, everyone gets it right. Everyone knows whether it's a red joker or a black joker, and there was just a hint, a bump of change in terms of reaction time. But finally, in this task, the subjects are looking at the whole deck of playing cards. So there's an ace of hearts, and then a 5 of spades, and a 3 of diamonds. And you were asked to remember what the last card was and say whether the next card is the same or different. And here what we found is that after the first dose, there's an improvement over placebo. And then as we dose through the 8 days of the clinical trial, every day that we do testing, the treated group is performing better than the placebo group. So everyone gets all the answers correct, and we're looking at reaction time, and we're actually seeing a 10% improvement in how quickly people are responding with correct responses. So think about in your daily life. This is about driving a car and recognizing who's turning left or whether there's a child riding a bicycle. It's about going to the store and remembering what list of items you're going to pick up at the store. It's about our tasks of daily living. So we're really encouraged by these early results. We're now moving the drug into phase 2 human clinical trials for Alzheimer's. We're targeting early Alzheimer's disease, where we think our drug has the best chance of showing benefit. And if it does show benefit in early Alzheimer's in terms of immediate cognitive improvement, we do think it has the potential to slow disease progression. And so, Dr. Fillit, thank you very much for the support by ADDF. ADDF co-funded this particular clinical trial with these exciting results.
Thanks to all the panelists. So now we have question and answers and discussion. And if any of you have any questions right now, please ask. Ronald. Oh, yeah, we need a microphone. The microphone, please, up here.
I had a discussion this morning with my brother Leonard, and the question we were discussing is when you have breast cancer, it's obvious. When you have diabetes, it's obvious. When you have heart disease, it's obvious. The question is, are we testing people with Alzheimer's or dementia? How can you tell, frankly, if somebody has early Alzheimer's, other than killing them and looking into their brain?
Yeah, actually, I thank you for that question. I would like to answer that if you don't mind, because I think it's a great question, and it actually well, it illustrates a great success of ADDF, and I don't think it was just a planted question, but it's a question that we get all the time. Oh, it was planted. Oh, okay. So, well, Leonard's right that in the old days, when I would see a patient with Alzheimer's disease or with memory problems, the only way I could tell them for sure if they had Alzheimer's disease was either a brain biopsy, which we never did, or an autopsy. And I've told this story many times, so you all might have heard it before, but it illustrates a lot of important things that you heard even from today from the panelists about biomarkers for clinical development. We got a call from someone at the University of Pennsylvania in 1999 who said, I have a great idea, I can't get any money for it, I want to take a molecule that Alzheimer used to stain the plaques at autopsy in a 52-year-old woman and showed that the woman had Alzheimer's disease, and I want to use it in a PET scan. And I proposed that by radiolabeling this molecule, it'll go up into the brain, it'll bind the plaques, and we could see it on a PET scan just in everyday clinical practice, and make a diagnosis of Alzheimer's disease in life without a brain biopsy or an autopsy, a definitive diagnosis, which was Leonard's question. And we supported that for several years at the University of Pennsylvania. In 2005, a biotech company called Avid Radiopharmaceuticals was spun out with $7 million in venture capital. In 2010, that company was sold to Eli Lilly for $800 million. And in 2012, that test was approved by the FDA as the first diagnostic test for Alzheimer's disease. And I have a small practice and I use it, and it's being used around the country. There's a study going on by Medicare right now of almost 19,000 people. We heard about the results of the first 4,000 people where dementia experts were diagnosing people in the community with Alzheimer's disease all around the country at something like 1,100 sites. And we looked at how, Medicare is looking at how accurate the doctors were in diagnosing Alzheimer's, and these were classified as dementia experts. And it turns out that doctors were accurate in diagnosing Alzheimer's disease clinically only 50% of the time. So 50% of the people who were diagnosed with Alzheimer's didn't have a positive brain scan and didn't have Alzheimer's, and 50%. So either they didn't have it when they were diagnosed it, or they did have it when they were not diagnosed it. More importantly, 70% of the people who had a positive scan had their clinical management changed. Either their medications were changed or their clinical management was changed. So Medicare is looking at this now to see what impact the scan has. But the reason we developed it is because we wanted to accelerate drug development. And what that means is that what we need in drug development, you've heard the 3 programs today, they're in phase 1. And you heard, for example, Mark showed you a PET scan of a biomarker of his drug, showing that it reached the human brain, it showed the distribution in the human brain, and he was able to use this in what basically would mount to the equivalent of what we call now a phase 0 study, something that we're very interested in, where we can use these biomarkers early in clinical development to see if they really work. Today, what's happened in the field of anti-amyloid therapy, because this is an anti-amyloid PET scan, is Lilly, during the progress of probably $1 billion program to test some anti-amyloid vaccines, went back and looked at how many people had actually, who were enrolled in these clinical trials, actually had Alzheimer's disease. And it turned out that around 30% of the people enrolled in the trials had negative scans. So what this meant was that Lilly was, and this was why Lilly bought the test, Lilly actually risked a lot of money on an anti-amyloid vaccine, went out and did this multi-center international trial, dementia experts enrolling people, and when you have 30% of people in the trial that don't even have the target that you're going after and didn't have Alzheimer's, it's going to mess up your results. You can't get positive results. And the most exciting thing that's happened in our field, I think recently, is about 2 years ago, Biogen, and this is why Biogen is so exciting right now, the results is that they used the scan to enroll, every person who was enrolled in the trial had to have a positive scan. And then the scan was used over time, and it was shown that 75% of the amyloid plaques were removed by the antibody, by the Biogen antibody. So the drug worked at 18 months. The drug showed efficacy. And there was some hint that the drug also improved clinical conditions or stabilized clinical conditions. And so Biogen went from a phase 1B, which you've heard about today, these are usually very small trials, 160 people, and risking almost today, as we heard an announcement today, they're risking another $500 million. So they went immediately from this phase 1B to a $3 billion program to develop the antibody. And really, when you think about it, what enabled that was really this biomarker. So with that being said, as an example, I just want to turn it over to our panel to talk about how important, if I could ask Leen, for example, how important biomarkers are going to be in the conduct of your phase 2 trials now that you're doing phase 1 for safety and maybe some little bit of efficacy, but could you talk about how important biomarkers are in clinical development?
So for us, we actually look at biomarkers in 2 angles. One is how can we use biomarkers to improve the design of our clinical trials. So we use it to stratify patients, as probably it's becoming a common knowledge that Alzheimer's is a complex disease, so there's multiple things that are happening. So in addition to the biomarker that Howard was describing, we've been looking at structural brain changes, so we're going to be using MRI, which again, ADDF was involved in the development of the Alzheimer's Disease Neuroimaging Initiative. So all of these things are important to help us select the patients, optimize the design of the clinical trials, and give us an early readout. Instead of waiting 3, 4, 5, or even more years for a patient to show a clinical change, we could have an early readout as early as phase 1. But all of us here today, in phase 1, we were able to include patients or elderly volunteers and use these biomarkers to help us guide future design and help us decide on doses and also decide on patients.
Yeah. Jerri, did you want to comment on biomarkers in your program?
Yeah, so I think that Howard and Leen both had excellent points in that you really need to have something to help you stratify your patient population because, as it's been mentioned, a significant number of patients don't progress to develop Alzheimer's disease, and they're enrolled in these clinical trials, and inevitably they end up resulting in a false or a failed trial. So for our clinical trial, what we've done is use an early biomarker of EEG to demonstrate alterations in brain activity with our drugs to give us a hint of some type of target engagement to ensure that our target's getting into the brain and having the effect that we wish to achieve. So those are the types of biomarkers that we're using in addition to looking to see if they have alterations in their brain activity that we would expect to see in an Alzheimer's patient.
Mark, would you comment on biomarkers in your phase 2 that you're hopefully going to fund soon?
It's amazing to me how much effort it was to make a PET ligand. So it took us 3 years of chemistry to develop our drug molecule, and then another year of preclinical tox and safety testing before we could enter human phase 1. It's taken another 3 years of chemistry to develop the PET ligand. And it's really, it's important in 2 ways. One, it's shown us where is the target in the brain, and guess what? It's where the brain learns and remembers. And now as a research tool, this will enable us to more broadly study this pathway, both in Alzheimer's disease, other dementias, and psychiatric disorders. And then from the drug discovery perspective, in phase 1, we're doing primarily safety studies and hoping for signs of efficacy either with a biomarker or in our case with a cognition test. But we're also trying to establish the maximum tolerability. So the safety studies are generally pushing dose as high as they can to decide, is this drug safe enough to take into a more impaired population? And so what the PET ligand will allow us to do in our trial is now understand as we back off from that maximum dose, where do we need to be in order to see the cognitive benefit?
And just to give a perspective on this, what you're hearing today is so different than what you might have heard 5 years ago and certainly 10 years ago in our field. When I started this in Alzheimer's research in 1980, there were no clinical trials. And we didn't know how to do clinical trials. So you heard from Ronald, we have 1,700, maybe altogether 3,000 clinical trials in cancer. But we've been doing clinical trials in cancer since the 1940s. So cancer field has learned how to do clinical trials, not just the basic science, but they've learned how to do clinical trials. We're just starting this really innovative stage of our work where we're incorporating these critical biomarkers into our work so that we can understand what the effect of our drugs are on human beings, which is really, Alzheimer's is a uniquely human disease. So we've had a lot of failures. But maybe the reason the drugs failed is not because the drugs didn't work, but like the Lilly example I gave you, maybe we didn't do the clinical trials right. Maybe we even need to go back to some of those drugs and look at how they might work when we're doing proper biomarker, using proper biomarkers and enrolling the right patients into our field. And I think it's a very exciting time that we've really upped the rigor and the effectiveness of the clinical trials that we're doing. I might add that both what Jerri's doing on M1 agonists, actually the validation for that program and our interest in it goes back a long way. There were major drug companies that tried to do this years ago and went into clinical trials and failed. And the advance of Jerri's work at Vanderbilt is not just in the unique chemistry that she's using, learning from the failures of drugs like xanomeline that failed because of side effects. Actually, xanomeline was an M1 agonist like Jerri's drug, and it actually showed efficacy in clinical trials, but it had terrible side effects. And so the chemistry is so amazing here because Jerri's team has been able to engineer out chemically some of the side effects. It's called a positive allosteric modulator. Did I get that right? Big word. But she's been able to engineer out this chemistry. And the same for Mark's drug. There was an old drug called Rolopram that never really went into humans because it had horrible side effects, and Mark's been able to engineer that out and show the efficacy. Let me turn to the audience again. Yes, Carolyn.
I was going to ask Dr. Rook, but also maybe Dr. Kawas, would your drug also enhance cognition in healthy people? And if so, I mean, do you have a safety profile that someday could help all of us?
It's an excellent question, and it's possible. What we do know is that our compound does enhance learning and memory in normal, healthy rodents. And so that's where we start, of course. They don't have to be impaired. We've tested them also in aged animals that have deficits or Alzheimer's disease models that have deficits. But our initial screening campaign was based on enhancing cognitive function in a normal, healthy animal. And so it's extremely interesting. It's not what we're targeting, but it is possible.
You know, I think part of that question also about healthy elderly, There are clinical trials going on of healthy elderly who have pathology, because we can find them in the community now with the scan, and part of the value of this amyloid scan I talked about earlier is that there are huge, very large clinical trials going on in normal, healthy people in their 50s and 60s who have positive scans but haven't got cognitive impairment. And I think, Carolyn, your question raises the issue of the separation between disease modification and symptomatic therapy. So we typically think of, for example, Aricept, and there's so much nihilism about the use of Aricept in the community that it doesn't work. But it actually does work. It actually works very well in about a third of people. But there's been and we think of it as a symptomatic therapy, but there's recently been some work published, and there's a fairly large body of work, that Aricept, even though we think of it as a symptomatic therapy, actually might be a disease-modifying therapy. And so recently there was structural MRI looking at the volume of the brain of the hippocampus where memory and learning is, and it's affected in Alzheimer's disease, and showing that people who take Aricept have slower atrophy rates in their brain. Structural change from the drug. They decline more slowly in their atrophy when they have Alzheimer's disease. And so, maybe one of the panelists might want to comment on this dichotomy that we tend to think about that might not be real, on the difference between symptomatic agents and disease-modifying agents. Leen, I don't know if you have any thoughts about
Yeah, I think that this is, it's a continuation of the same question. There are two lines of drug development. One symptomatic that is similar to the NSAIDs, like basically ibuprofens that you guys take. It doesn't cure or addresses the underlying causes of the disease. And there's the other line, which I think most of us are trying to focus on, is changing the disease or the root cause of the disease. For us, we're targeting neurodegeneration, so the dying of the brain cell, the atrophy process that Howard was describing, and we're trying to recreate the lost connection, the synapses. So this potentially could cover a wide range for patients. A lot of the technologies right now are trying to go prodromal, even before when you actually diagnose someone with Alzheimer's. So there is a push that we need to start looking at it earlier, so we try to combat some of the changes that happens, whether it's the protein pathologies or brain cells that are undergoing the death sequence and losing the brain matter. So this is a very critical point, and this is where the field is moving, is we understand now there's so many causes of the disease: inflammation, degeneration, the protein pathologies, and we need to address all of these either together or whatever is most presented in the patient population that we're working with.
I think that's a really important point to make, what Leen said, and one of the reasons we're so interested in her project, because we know now from the biology of Alzheimer's that there's so many different ways that neurons can get injured: inflammation, oxidation, vascular hypoxia, low oxygen, and so on, neuronal energy failure. And so there's so many ways that neurons can get injured, and we're trying to treat each one of those ways. But down the funnel, there's something called neuroprotection. And so these are pathways in the cell that protect the neurons from dying. And the signaling is either, okay, we're so sick we have to die and go away, or we have a chance to survive and we're going to neuroprotect ourselves. And so what Leen is working on is called growth factors, that are these neuroprotective pathways that can protect against cellular injury regardless of the cause. And so this would be really a parsimonious approach to treatment. And it's one of several programs we have in this field of neuroprotection. And Leen has also called her program regenerative, which I think is a great word. And I wonder if you want to comment on why you think your growth factor approach to Alzheimer's is also a regenerative approach to the disease.
So part of the work that was funded by the ADDF, again, it's still preclinical data, but basically the outcome of activating these growth factors, think about it as a switch. When you switch it on, basically it starts a regenerative process. Alzheimer's and other neurodegenerative diseases, basically the core cause for the clinical presentation is the degenerative process, when you lose the brain matter or the connectivity, the synapses that we've been discussing earlier this morning. So what we're doing is we're switching on this factor in the brain, which actually recreates synapses. So we've seen preclinically that the synapses that we've seen earlier in the video that Howard showed, they're being recreated and they are functional. So we were able to detect enhanced brain activity. So we're not only changing the brain structurally, we're actually are able to enhance its functionality in a very positive way, which we hope it's going to translate in the clinic. We're using the same biomarkers as well in the clinic.
Any other questions? Yes?
Good morning. My name's Lorene Arbus, and my question is, there's a great deal of controversy around a particular process called chelation, which addresses, for those who believe in it, the loss of memory and brain fog by removing toxicity from mercury and other of the 20 or so metals that are tested, more than 20. Could you comment on the present state of point of view regarding toxicity?
Yeah, maybe I'll comment on that as a clinician. There was a time when we thought aluminum, for example, was related to Alzheimer's, and that was proven to be definitely false. There is some role for iron in the pathology of Alzheimer's disease. Mercury, certainly, and lead can be risk factors for brain degeneration when they're around in excess. I think the question clinically about chelation is really how it's used. And I'm pretty much a traditional doctor, and I would say that if I saw someone, for example, that was iron deficient, and it's quite common in the population, I would supplement them with iron, and iron deficiency, as we all know, causes anemia. B12 deficiency causes neuropathy and can cause Alzheimer's disease, so we always, can cause cognitive impairment, and we always measure B12 levels in patients who come into the clinic with memory problems. The issue with chelation is that it presumes that there's an excess of these compounds, and so we need to measure it. And in people who have an excess, we need to find out why there's an excess. And I just worry that the practice of chelation therapy is not doing it in a way that's really related to actual blood levels of these metals. And with regard to Alzheimer's disease, I think there's probably no evidence that chelation therapy, which I know has been tried, desferoxamine being an iron chelator that's been tried, there's no evidence that chelation therapy works for Alzheimer's disease. In fact, the whole issue of supplements in Alzheimer's disease was raised by the question, I think, and we recognized several years ago that, and I like to tell the story, not too long ago, I saw a patient who, a former ambassador of the United States, who we diagnosed with Alzheimer's, and while his wife was there, she said, "Can I come see you?" And I said, "Sure." And she came back a few weeks later, and she had a carry-on bag with her. And I said, "Oh, where are you traveling?" And she said, "Oh, no, you told me to bring in all my medicines and supplements, and they're in my carry-on bag." And so we opened up her carry-on bag, and she laid them out. There were literally 30 or 40 supplements that this lady was taking, prescribed by various nutritionists and doctors and so on. It's that kind of patient that we wanted to create a place that people could go for credible, understandable, evidence-based scientific information on the use of supplements and other medicines for Alzheimer's disease. And we do have a website that I'm very proud of. It's been built by our staff, called cognitivevitality.org. And if you have any questions about supplements, curcumin, turmeric, ashwagandha, B vitamins, whatever question you might have, I think you'll find reports on there that are very well written. We're very lucky to be supported by an anonymous donor who is helping us with this website, and we're expanding its distribution. And so if you have any questions about supplements and those sorts of things, please visit Cognitive Vitality. Any other questions? Yes, sir.
I just have a question about the M1. Is there any incidental evidence that if you were able to like take tremendous amounts of Aricept without side effects that you're working on that would show that there would be a beneficial clinical result? That because you're saying because of the fact that you can't do it without the side effects is the premise, but what is the thought that is there any incidental examples or data that showed that if you were able to load somebody up with that drug without the side effects that there would be a better benefit?
To my knowledge, and Howard may be able to help me with this with his clinical experience, the side effects are so severe. First of all, they begin by titrating. You start with a very low dose and slowly titrate up that dose as high as you can, with the hope that they're going to develop a little bit of tolerance to those adverse effects. But generally speaking, because you're activating all of those receptors, I don't think you're going to be able to get to a dose that's high enough to activate the M1 receptor with enough effect to have efficacy without having severe side effects in the outside of the brain.
As you titrate up, as you increase the dose of these drugs, the toxicity is lethal, essentially. So it's a bad side effect. Yeah.
It's an excellent question, though.
Any other questions from the audience? Yes, one in the back.
What is the percentage of children with one Alzheimer's patient getting the disease, and do you think within the next 15 years there will be a vaccine or a preventative that people can take before they show any symptoms?
Yeah, the genetics of Alzheimer's disease is quite interesting. We've had a gene that we've known about for Alzheimer's disease called apolipoprotein E. We've known about this gene for over 25 years. And people, there's 3 different types, 2, 3, and 4. And people who have the 4 type, if they have 2 4s, one from mom and one from dad, they have 15 to 20 times the risk of getting Alzheimer's disease. And it's even been shown that children that have ApoE4 have altered connectivity in their brain. So there's something very fundamental about this gene. It's not a mutation in the sense that it causes Alzheimer's, and not everybody that has the gene gets Alzheimer's in late life, but it's a genetic risk factor. And what I'd like to pull that question into today's discussion is, how do genes inform us about how to develop drugs for people with Alzheimer's disease? And the challenge has been that we've had a portfolio of trying to fix this ApoE4 gene risk factor, genetic risk factor, for probably almost 15 years now. And it's been very difficult because we can't quite figure out exactly how it relates to Alzheimer's. So what we're doing now at Cornell here in the city with Dr. Ron Kriskell and Dr. Greg Pexko and the support of some of our donors in the audience is we're doing gene therapy for Alzheimer's disease. And the way this is going to work is that we're able to take a non-infectious virus, a virus that doesn't cause disease but can transmit DNA into the brain. And the protective type of ApoE is called ApoE2, and we know from animal models that if a mouse is 2-4, or even in people who are 2-4, the 2 offsets the 4. And so we're going to be studying ApoE4 people and giving them the ApoE2 gene in a virus so that these people who have ApoE4 will start producing ApoE2. And we're very excited about it. And we're in this IND enabling stage right now, where we're bringing that virus directed gene therapy for this genetic risk factor into first-in-human trials for the first time, and we're very excited about it. So eventually, it might be that children that have ApoE4, or maybe middle-aged adults that have ApoE4, would get this vaccine. They might just need 1 shot every 5 or 10 years, and it would protect them against Alzheimer's disease. The other forms of Alzheimer's that are really mutations that cause Alzheimer's are quite rare, and they're very informative to us in terms of maybe how to develop the vaccine, and there are clinical trials going on with that right now. But I think most of us don't think that everything that's going on in the rare genetic forms, less than 5% of the population, will really be ultimately necessarily useful in the vast majority of the other 95% of people. So
risk value if one parent has Alzheimer's, and how long do you think it'll be before middle-aged children can get a vaccine to prevent it?
Yeah, so I'm going to go back to the scan and how this scan has really changed the world. And actually, there was just an editorial in the journal Neurology, which I just want to say in a humble way that has really said that this technology has really revolutionized Alzheimer's research and care, is that because we can do these scans now in 40 and 50 and 60-year-old people before they have any symptoms, it would be like if somebody just had a high cholesterol and maybe they had some atherosclerosis in their coronary arteries, but they haven't had a heart attack yet, so they haven't had the real disease expression yet. And we can treat those people and prevent heart attacks. We're doing that now in Alzheimer's disease with a vaccine where we're finding people in the community who are essentially normal on their cognitive function. We're scanning them, we're finding those people that have amyloid in their brain, and then they're being treated with a vaccine. So those trials are going on right now, and I think that we'll know the results from those particular trials within 5 years for sure. So if those trials work, we'll have the first preventative therapy for Alzheimer's, and I think there's many, many more to come. Any other questions? Okay. I must have said something. Some more questions here? Okay.
I recently read a book by a Dr. Dale Bredesen called The End of Alzheimer's, in which he claims to have eliminated Alzheimer's from a number of his patients. Do you have any, it seems to have impressed a number of doctors.
Yeah.
You have any comment about it?
I don't have any comment.
I'm sorry?
I think we know Dale. Actually, we funded Dale when he was president of the Buck Institute about 10 years ago. I think the best I can say is that I think he's promoting good clinical care, and something that all doctors should be doing to take care of Alzheimer's patients and make a good diagnosis. Some of the things that I think he's proposing are either not really feasible, or he's proposed some supplements that I don't think there's necessarily good data on. Some of the supplements we actually have reports on in our Cognitive Vitality website, where we review all the evidence in an understandable way. So I think the best thing that people can do if they want to prevent and treat Alzheimer's is to work with a physician who's well informed in the care and diagnosis of Alzheimer's disease. Some questions up in front here, I think.
I just had a quick question for the panelists. How are your drugs administered? Are they oral? Are they IV?
Our drug is oral.
They're all oral.
For us, it's sub-Q like insulin, so you could take it at home. It's not an IV.
Any other questions? In the back.
Thank you. To what degree is the R&D that the fund has been involved in, or the panel may want to address this, to what degree is it relevant to the concussion-related dementias that we see versus true genetic Alzheimer's?
That's a great question. Leen, you?
Yeah, so that's actually in line with what Howard was describing earlier, is our technology does not basically addresses whether it's traumatic brain injury that resulted into a degenerative process that led to dementia and Alzheimer's, or if it's basically the cardiovascular system is not functioning well, not enough blood is going into the brain, so there is a degenerative process that leads to cognitive decline, or if it's the typical, inflammation, protein pathology that also leads to the funnels into the degenerative process. So for our technology, we do see potential that it would expand and cover, universally any type of cognitive decline that's connected to, and most of them, if not all of them, are connected to the degenerative process. Even normative aging, even normative healthy aged people do have a change in atrophy and losing the connection to a certain level. There's a threshold that after that you become clinically diagnosed as either demented or Alzheimer's. So we do see, and this is why I think that Alzheimer's Drug Discovery Foundation has been excited, because there's potentially an expansion to not only one type of Alzheimer's, it could expand to other causes of cognitive decline and potentially other neurodegenerative diseases. So potentially could expand to other Parkinson's and others that have similar and overlap pathologies to Alzheimer's.
Mark?
Yeah, thanks. We've been working extensively in the area of traumatic brain injury. Our interest has been in the chronic phase of injury. So what happens to patients 1 year after injury, 3 years after injury? There's about 1.2, 1.5 million concussions per year, and 4 million Americans with permanent disability after a TBI. So we've been working with the Miami project to study animals in this chronic phase. So we asked them, our collaborators, Dalton Dietrich and Colleen Atkins, do you have a model where you injure a rat and you wait, and then you start treatment? And we've been very impressed and pleased by the results that we found in that collaboration. So this pathway that I described to you, the PKA-CREB pathway, is impacted by TBI. And when we test our drugs in these animal models, rats 3 months after injury with no earlier treatment, we see recovery. We see the animals now performing in a variety of tasks of learning and memory at the same level as sham-injured rats. So we're really excited by these data, and that's a parallel program for us at Tetra.
And the ADDF is also interested in related dementias, and chronic traumatic encephalopathy is certainly a big problem. And we are funding some programs. The people with chronic traumatic encephalopathy get tangles, and that's a post-concussion syndrome. So the way we look at it from a drug development point of view is that by funding drug development for chronic traumatic encephalopathy, which is a disease that primarily has tangles, if we can learn how to treat a pure tangle disease, we'd be able to apply that technology to Alzheimer's disease, because people with Alzheimer's disease also get these tangles that represent the dying neurons. And we're funding some work at Mount Sinai right now with Sam Gandy on using imaging, some novel imaging techniques, because the next exciting thing you're going to start hearing about is not only about this PET amyloid scan as a biomarker for diagnosis in the clinic and to accelerate clinical trials, but you're going to start hearing about PET scans that can see the tangles. And that's really exciting, because the thing about amyloid scanning and amyloid deposition is that it doesn't actually correlate with cognitive function over time, but the tangles do, and that was shown on pathology by Brack and Brack about 25 years ago. And they showed that as the tangles, as the dying neurons spread through the brain, and you can see them at autopsy, it's a direct correlation of those dying neurons or those tangles with cognitive function. And now we have brain scans, and there's a number of them in development by imaging companies, where we can actually do that same injection of a radio-labeled molecule, and goes up into the brain, it binds the tangles. And so that's going to be really important, because it correlates with cognitive function. It'll be a surrogate for not only early diagnosis, but in clinical trials, now we'll have a scan, because cognition, as one of the problems in our clinical trials, and again, I go back to this theme of that we're learning how to do clinical trials better. Cognition is a very variable outcome measure. It has a lot of variability in it, both from one site to another, from one neuropsychologist to another, from one patient to another, from one day to another. But the tangles that cause impaired cognition don't vary. And so they're a much more objective, clean measure of the efficacy of a drug, because they measure whether the drug is preventing neuronal cell death, and they're also telling us a correlation between a drug and how well people are going to think. So you're going to hear a lot about tau, what's called tau imaging, PET tau imaging, as we go forward. And we're supporting, in collaboration with Merck at Harvard, the development and the widespread use of this tau imaging technology, again, to advance the efficacy and the rigor of clinical trials as we go forward. I think we have time for maybe one more question from the audience, if there is one more. Okay, well, I want to thank our panel, terrific panel today, for their involvement. And we want to thank you for your work. One of the things I had hoped to get into was your personal stories about why you are doing this horrible, difficult thing with your lives. But I think that the reason is that we all enjoy science, and science is the creation of new knowledge, and I think we're all creating new knowledge, and ultimately we are going to get to a drug. So we're very excited about that. And I want to thank all of you for joining us today and your attention. And let's retreat to the ballroom for lunch. And thank you so much for coming. We really appreciate it.